So today again we are going to continue discussion about the forward genetics, and just to have a quick recap what we were talking about. So we were talking about setting up a forward genetic screens and some of the questions a geneticist may be interested in before he thinks of setting up a genetic screen. For example, you may be interested in amino acid biosynthesis. You may be interested in disease resistance in plants. You may be interested in process of transcription, how genes are regulated. You may be interested in how host and pathogen they interact when a disease onset takes place. So the questions are, there's an unending list of questions. And last time we talked about the selection of the mutagen is very important. So which mutagen you should use, as well as what concentration of or the dosage of mutagen should be used. So we went through these steps, and we talked about different questions you may be interested in. For example, it is written resistance to antibiotics, resistance to various inhibitors or pathogens, etc. You may be working with bacteria, so you want to learn about how bacteria they resist viruses or then herbicide resistance. I told you how you do these screens. For example, here it is amino acid biosynthesis screen done in Neurospora carasa. We talked about how you can design an assay. So this is a selection assay. Last time I explained you how amino acid biosynthesis mutation screens, they are the assays normally screening them on minimal media plus specific amino acids for which you are screening for. This is a fly screen where they have screened for phototaxis or behavior of fly towards light, why they are always attracted towards white, and what could be the genetic basis of this phototaxis in flies. And then this is a selection assay. Last time we talked about this is, a T-shaped thing. On one side you have light, on the other side you don't have. When you will pass the mutant population, so if there are phototaxis defective flies, they will move towards dark side, and the wild type will still, the ones which are not mutant in the phototaxis genes, they will go towards light. As compared to the wild type will always go towards light. Similarly, you can do other things as well, olfaction. You can think of, taste, smell, sweet and bitter. You can have, on a plate, you can, on one side, you can add food or sugar source. On other side, you can have, for example, something extremely bitter. You can have hydroxychloroquine or the malarial drug, which is called quinine. You can add here, and in the middle, you can, leave all the larvae, and the the ones who will be defective in taste, they will go on this side as well, and the ones who know that sugar is our source, they will always go towards sugar. So these are different kinds of assays you develop to do a genetic screen. This is something very interesting. I recommended you last time that you should go and read about this screen, Leland Hartwell and the Paul Nurse screen, which led to discovery of cell cycle mutants. Normally, you cannot do such a screen because if you have one cell, which normally after cell division goes to two cells, and we know this happens through today in 21st century, we know it happens through, going through the cell cycle, G1, S, G2, M. So this whole cell cycle we owe to discoveries by Hartwell and Paul Nurse screens. Now, why it is extremely genius assay? Because not if you have mutations in genes which are responsible for this process, going from mother to two daughter cells, the mutants will die. So they created temperature sensitive mutations, and temperature sensitive mutation made their jobs easy, because normally on normal temperature yeast grew very happily, but when they raised temperature, they could see all different kinds of phenotypes in the mutants, which are shown here, and we also discussed them last time. Based on all their discovery, the whole cell cycle, the genes which are involved in cell cycle, cyclins, and then the cyclin-dependent kinases, this whole cell cycle was characterized based on the genes which were discovered in cell cycle screen, and we could clearly see how cell goes from one cell to two daughter cells after cell division. One of the landmark discoveries through genetics was understanding the segmentation process in fruit flies. It was led by Christian Noslin-Follard and Eric Weishaupt. They both in their postdocs, I think in 1970s, early 70s, they were interested in understanding how the segmented body plan is established in fruit flies. For example, fruit fly is divided in head, then this thorax region, and then this abdomen region, okay? In these segments, and the segmented body plan is established early during development. So this is 3 hours old embryo. So after fertilization, so the egg cell is laid by mother, and all the development takes place outside mother's womb in fruit flies. After 10 hours, you see here already, so there are segments visible. And if you pay attention here, it is written T1, T2, T3, and then there are A1 to A8. So total, there are 14 parasegments. So at this early stage, the fly body is divided into, or these segments are called parasegments. This division of body is maintained. These segments are retained in the next stage, which is the larva. So from 0 up to here is 24 hours. And then, this is 10 days. At 10 days, the adult hatches. After, 5 to 6 days after egg laying, the larva, after passing through 3 different stages, goes into pupal stage, which is a sessile, immobile stage within a hard casing. And from pupa, in next 5 days, you have these adults. Now, the segmental body plan, which was established early during development, this is maintained during rest of the development. And if you change segmental identities, you can die, you can not survive, or you may have very severe morphological defects. For example, we talked about bithorax phenotype. We talked about antennapedia phenotypes in the introductory slides. That is disturbing the segmental boundaries. Now, these two scientists, Kristen and Eric. Eric is now professor at Princeton, and Kristen was last time when I saw her, she was director of Max Planck Institute in Germany. So using classical genetics approach, what they did, they simply mutagenized flies using EMS, and they took males, they fed males with the EMS mixed in sugar, and then crossed with, these males were simply crossed with normal wild type females, and in the progeny they look for any segmentation defects. This screen was so successful that almost every gene which we work on in our labs has, one way or the other, it goes back to this screen. Classical genetic approach, and they screen for phenotypes where these segments were defects. For example, based on their discovery, so we will talk about these landmark discoveries in few lectures from now, but based on their discovery, what they discovered that, there are 5 different classes of mutants. So they grouped the mutants in 5 different classes based on the phenotypes. They named maternal group genes, then the gap gene mutants, pair rule gene mutants, segmentation genes, one is missing here, and then homeotic genes. Maternal genes are the ones what they observed that, if mothers carry the mutation, then we see phenotype. If mutant mothers are crossed to normal wild type males, we see mutant phenotype. But if normal wild type mother is crossed to mutant males in the same gene, we don't see phenotype. So they named them maternal genes because they thought mother is contributing. These genes are extremely important during egg laying or during gametogenesis, that if they are mutant, the segmentation patterns are disturbed. And 2 examples are Bickert and Cordell. Then they named gap genes. We will talk about all of them in 2-3 lectures later. Gap gene mutants were, if this class of genes were mutated, there were big gaps in the mutant embryo. So these segmentation patterns are, some big gaps are created or produced in subsequent development of fruit fly. Next class was pair rule genes, and pair rule genes were the ones which were expressed in alternate segments of early stage embryo. And then was the segmentation gene. Segmentation gene were the class of genes which also result in defects. And finally, the homeotic genes, they were later on also termed as master regulators of development. And these master regulators of development, if you have mutations in homeotic genes, what happens, you have, for example, T3 segment, it becomes T2, or let's say T2 becomes T3, or like we saw Antennapedia, so legs on the head, legs normally develop here in the T2 segment, so which means head region has become T2. So the word homeotic is from homeosis. Homeosis means conversion of one body segment into another body segment. And then finally, the polycomb trithorax, these are epigenetic regulators. these are the ones which bind to the chromatin at specific sites in the genome, and their job is to maintain the body pattern. They maintain the cell fates. So what has happened when you were here, cell fates were not yet established. But as we go from simple development towards more complex, what is happening here somewhere, cell fate determination is taking place. Fate of different cell types is being established, and once they are determined, then through mitosis, through subsequent cell divisions, it is being passed on to the later stages of development, and eventually, Polycomb trithorax, they take over the job and they maintain the cell fates which were established early during development. How do we do these screens? Whether EMS is and MMS is the only way to do these screens, or there are some other methods as well. So 30 years or so, or a bit more, in fruit flies, they also discovered a transposon, which was named as P element, and the P element contains, repeats on both sides, and then they have a transposase gene. What Drosophila scientists did, they removed all the transposase gene from here. They kept the repeats here, the borders, and replaced it with some, let's say this region was replaced with mini white gene, the gene which gives, red pigment. You can also think of having GFP or whatever other easily selectable gene. And then this whole, after removing transposase, they put it on a different plasmid. And here, what they did, they cloned their gene of interest or whatever they wanted. And you can inject the transposase helper plasmid together with your P element construct in embryos, and you can create transgenic flies. And this P element, since it integrates randomly into the genome, it's basically creating mutations as well. So P element mediated mutagenesis is also done a lot in last 30 years. In Arabidopsis, people have used EMS, MMS, and it will be unfair, and I will not do justice with functional genomics and genetics if I don't talk about plants. Without historic forward genetic screens and plants, which have contributed enormously to understanding of fundamental cell and molecular biology, without Arabidopsis or plant screens, this was not possible. Similarly, in addition to EMS, MMS screens in Arabidopsis, tDNA is basically so Ali Heather, do you know what is tDNA? Ali Heather?
Yes, sir.
So what is tDNA?
सर जो जिसको हम ट्रांसफर कर रहे थे
G Hamza Aslam, what is tDNA?
सर tDNA जिसमें ट्रांसपोजेज जीन होगी
Sadiq Ali?
वो
Sir, it is basically the transfer DNA, but it is causing the tumor in agrobacterium tumor in the plants.
Yeah.
It is,
So how it causes tumor in agrobacterium?
The genes which are it causes tumor in plants, and the gene is from Agrobacterium tumefaciens.
Yeah.
So
What is the mechanism? How it causes tumor? Why it causes tumor?
so that the bacteria can get the nutrition from the plants.
Amna Mahfouz, how does it cause tumor in plants?
Sir, I don't know the exact mechanism.
Navera?
सर जब भी किसी प्लांट के किसी पार्ट को कोई वूंड होता है ना, तो वहाँ से एक केमिकल क्रिएट होता है सीटोसिरिंजोन, जो कि बैक्टीरिया को अट्रैक्ट करता है बाय कीमोटैक्सिस, और बैक्टीरिया वहाँ पर जाकर अपना टीडीएनए रीजन इंट्रोड्यूस कराता है जिसके अंदर विरुलेंस जीन्स होते हैं, और वो विरुलेंस जीन्स फिर प्लांट के अंदर क्राउन गाल डिजीज कॉज करते हैं, और हम उन जीन्स को रिप्लेस कर सकते हैं विद आवर जीन्स ऑफ इंटरेस्ट टू गेट एनी डिजायर्ड ट्रेट इन प्लांट्स.
So what are those genes?
विरुलेंस जीन्स, विरबॉक्स.
Yes. What are those genes?
I thought these were virulence genes.
So virulence genes 1, 2, 3, 4 कोई नाम बताओ.
Virulence A, vir A है, vir G है, vir E है.
So what do they do?
Sir, they guide the tDNA to reach into their host safely and cause the disease.
ठीक है. So I wanted all of you to go and read about Agrobacterium tumefaciens. It's called TI plasmid, okay, tumor-inducing plasmid. It's as I think Novera was talking about. There are Vir genes. There is this tDNA region. Not the whole plasmid is transferred. So go back and read about tDNA. So it was not only used to TI plasmid of bacteria is not only used to create transgenic plants, but it was also used to create mutant screens and mutant population in different mutant screens, because tDNA also goes and integrate into the genome randomly. And random integration means if there are 4 chromosomes in Arabidopsis, 4 pairs of chromosomes in Arabidopsis, tDNA will go and it can integrate anywhere in the genome and create mutation, insertional mutagenesis through insertional mutagenesis in promoter regions. Mostly it lands in the promoter region, but it can land within the gene as well. And you have a mutant population which you can screen. So you should go and read about, as I said, TI plasmid system for Agrobacterium. It's nearly 200 kb. It's used to generate transgenic plants. The genes are like, one of the genes is nopalin synthesis. These genes basically induce cell division, like cytokinins and auxins, etc. And that's why you see tumor in Arabidopsis. So you should go back and read about how transgenic plants are produced in Arabidopsis, and also the mutant screens done in Arabidopsis. Using tDNA. Must read, okay? So like here in Arabidopsis, in Drosophila, we replaced these non-essential transposase gene with mini-white and genes of your interest. You do same things here. You remove these nopalin synthase and tumor producing Vir genes, etc. And what you do, you can think of in cloning GFP gene or antibiotic resistance gene, hygromycin gene, or kanamycin, whatever. And then you add this into plant tissue. For example, you simply take a leaf disc, mix with agrobacterium, which contains your gene, your plasmid now. We call them tDNA plasmids. tDNA plasmids are engineered plasmids. They are no more TI plasmids, okay? Which contain your genes of interest, and then a lot of these non-essential genes, they are removed, which may be dangerous. So remember, tDNA is not a transposon plasmid. This is very important to know. It's not at all a transposon. So P element is a transposon. Okay? And once you will infect these leaf discs, you can select them on, let's say you have hygromycin you're using as a selectable marker. So anything, if the leaf disc is transformed by agrobacterium, now on the plates you can have hygromycin, and all the discs which will show resistance to hygromycin means agrobacterium has transferred its tDNA in this, and your genes of interest are also part of it. And you play with different nutrient conditions, you play with auxins and cytokinins, rooting hormones, naphthalene acetic acid, etc. And from this leaf disc, you induce calli, and eventually the whole plant is made. This is what the process of creating a transgenic plants is shown. So you have, the co-integrate plasmid. So these are the tDNA left and right borders. Your genes of interest are there. Then the helper plasmid is there, which is going to cut the tDNA at this side. It will go inside the plant, get integrated into the genome of the leaf disc or whatever tissue you used, and you select them on whatever selectable marker, hygromycin or GFP or whatever you use, and eventually you induce rooting and shoots and have that transgenic plant. Since this tDNA gets randomly integrated into the genome, you have mutant population. You can create mutant population. You can use same tDNA to create transgenic plants of your interest. For example, you can clone gene of your interest and transform these plants, and then those transgenic plants will contain specific traits, specific characters which you wanted to introduce into the plants. For example, you are interested in creating bacterial resistance in plants, or viral resistant crops you want to produce, or you want to have herbicide resistant plants. So you will clone those genes, integrate here, and then you select your transgenic plants. That's the other use of this agrobacterium mediated transformation. Okay. This is what I already told you. So out of this plasmid, not everything is transferred, only the transferred DNA region is transferred, which goes integrated. And then eventually because in the first, what we call the T0 generation, which is selected here. So this is the T0. You cross them, and after selfing, 1/4 will show you homozygous, half will be heterozygous, and 1/4 will be wild type. And you go on and try to make your homozygous plants. In Drosophila, so I come to this slide later on. First, it's important to go here. In Drosophila, what we do, we have this P element, and as I told you earlier, the transposase gene is removed. You have these left inverted repeats on both ends of the transposase. This transposase gene is replaced with minivide. Minivide gene, when it gets expressed, it will give you orange colored eyes. Eyes will become orange, slight orange. We call this minivide because it's engineered. So white gene, which normally give you brick red eyes, it's engineered in such a way that you have a small gene which is still able to give you dark orange eye color. So you clone minivide as well as any other gene of interest. You have this P element plasmid injected together with the transposase, because you don't want to have transposase here. If it will be integrated, it will not allow your gene minivide and whatever your gene of interest is to be integrated stably, because transposase will again and again act and remove it, excise it from the genome. So you inject flies, embryos. Within first 30 minutes of egg laying, you collect embryos, and through very fine needle, you inject. What will happen, since it's transposase, so the transposase will act on left and this inverted repeat borders, and your gene of interest will go and integrate into the genome randomly. Let's say I'm showing it with minivide is on your gene. It can integrate here. It can integrate on any of the 4 chromosomes in fruit fly. What do? You take these flies and you cross them. Let's say this is T0. You now crossed it with another male, or based on if this is female or male, whatever. You cross them with the opposite sex, and then you select the progeny. What is important, I should highlight, since you are using minivide, it means the embryos which you will inject, they should be white minus. Their eyes should be white. They should not be wild type flies. So white minus flies, normally the ones which we use in the labs is called white 1118 allele. Okay. So after injection, we expect that when minivide will successfully get integrated, the progeny of this fly will be showing you red eyes. Remember, these embryos, when they will go through their life cycle and adult will hatch, so what will be the eye color of this if minivide integrated? So what will be the eye color?
Red.
G. Others?
Sir, orange.
Orange. Novera?
I think orange.
Sadiq Ali?
It will be orange.
Amna?
Sir, the minivide wasn't injected in the eggs, so shouldn't it be white?
Why white?
Because the gene was inserted in the germ cells
Yes.
of the embryo.
Yeah.
So the embryo itself should be white.
Good. Very good. So as Amna is saying, we have injected in the germ cells. And when this embryo will develop, all its other cells and nuclei, they are normal. And this generation will show simple white eyes, no orange or red. When this will be crossed, all the embryos which we injected and adults hatch, we cross them with white 11, 18, males or females, and we select progeny. Here are the chances if our microinjection was successful. So we will see in the progeny orange eyes. If transformation was successful. And then we crossed and make homozygous these transgenic flies. So we injected the germ cells. And germ cells in fruit flies, they are separated from rest of the body, like here in the posterior pole. They are called pole cells. All the other cells in embryo here, they are not injected with our construct. When this embryo will go through larval stages, pupal stage, and adult will hatch, its eyes will be simple eyes of white 11, 18, not red due to the mini white which is here, because only the germ cells are targeted. When we will cross this adult with again white 11, 18 in here in the progeny, chances are we see orange-eyed flies, because the germ cells which are injected here, they will be passed on through gametes. Fertilization will take place, and only in the progeny we will see orange eyes. Is it clear?
सर ये इंजेक्शन से हेट्रोजाइगोसिटी आई है बस हमारी फर्स्ट प्रोजेनी के अंदर, ये जो ग्रो किए हमारे यहाँ वाले लावर्स हेट्रोजाइगस हो गया और फिर नेक्स्ट जनरेशन में जाके उन्होंने अपीयर होना है एज
फेनोटाइप. Here it will be heterozygous in F1, not here. यहाँ पे तो ये आपने एम्ब्रियो को इंजेक्ट किए, ठीक है, इसके बाकी जितने सेल्स हैं उनमें तो आपका ये कॉन्स्ट्रक्ट नहीं गया माइक्रो इंजेक्शन वाला. इसके जब गैमीट्स बनेंगे, लेट्स से स्पर्म्स, वो जब फर्टिलाइज करेंगे, अगर तो ये इंजेक्ट हुए हुए तो फिर आपको प्रोजेनी में रेड आइज नजर आएंगी. क्लियर?
जी सर, यानी कि अगर हम इस तरह की इंजेक्शन करेंगे वो आइज डेवलपमेंट वाले सेल्स को जाके अटैक करे तो फिर आई में फर्स्ट टाइम ही कलर आ जाएगा. अब हमने इस वक्त जर्म सेल्स को किया इसलिए वो पहली जनरेशन में नजर नहीं आया.
Yes. So you create mutation. Yeah, this was important. Now, why I explained you in detail the fly transgenesis, because I want you to learn another forward genetic screen, very successful one in fruit flies, and that was called enhancer trap screens or promoter trap screens. For example, we are interested in eye-specific genes. Genes which specifically are expressed in eyes only. So the genetic approach will be that if we know the regulatory regions of genes, eye-specific genes, we will know all the genes which are expressed in eyes. Because when we say eye-specific genes, it means these genes are very specifically expressed in eyes. And you know, gene expression is regulated by specific cis regulatory regions, which are called promoters, enhancers, etc. And they normally enhancers. So I just draw the normal gene. Let's say this is the promoter of the gene. Then from here to here, we have the coding region or introns, exons, etc., the gene body. We have transcription terminator, etc. And upstream of promoter, so these hash lines means it can be very far, okay, or it can be next to promoter as well. There are regulatory regions which are called enhancers. Enhancers actually act on the ribosome, the RNA Pol2 machinery. They facilitate for activation of gene transcription. Enhancers can be upstream. They can be here within a gene, within an intron, or they can be downstream. Okay, not necessarily always upstream. They can be anywhere in the gene next to gene. So since their job is to regulate gene transcription, idea was that if we can clone enhancers, we will be able to understand, you know, tissue specific genes, eye specific genes, or wing specific genes, leg specific genes, if you go in mouse, heart specific genes. So geneticists, they designed a beautiful approach which we call enhancer trap screens. And idea was that we will have a p element construct. Okay. Now this p element construct, let's say we stick with mini white gene. Or we can think of lag-Z, bacterial lag-Z gene. Or people have also used GFP, mini white lag-Z, you can think of using anything. It contained a minimal promoter. Minimal promoter means it is just data box. It cannot, RNA Pol2 may bind there, but it cannot transcribe this at its own. So idea was, in the presence of minimal promoter, our reporter gene, which is lag-Z, GFP, or mini white, whatever, it will not be able to transcribe, it will not be active. However, if it lands through p element mediated transformation, if this construct, it lands somewhere near an enhancer, for example, it goes and integrate here. Okay. Now it's close to enhancers. The enhancer will act on the minimal promoter in our construct, and then our reporter will be active. Is it clear? Yes or no? Are you with me all?
Yes, sir.
Is it clear so far?
Yes, sir.
Good. So it means due to integration of our construct in the vicinity of enhancers, we will now see lag-Z or GFP or mini white. And people did, you know, such p element mediated mutagenesis. Sorry. Then they made this mutant population, because this is a mutant, you know. And in the F1, they went in F1, then F2 try to make it homozygous. It can be lethal as well, because, you know, if it is disrupting some essential gene here on which normally enhancer acts, you know, it may not be homozygous. You may have to keep it as heterozygous. Now, this enhancer can also act on this. So we are soon going to see. So you have, let's say, you created 5,000 transgenic flies, which are through enhancer trap. Enhancer trap. We call it trap because we are like trapping an enhancer to act on me, on my construct. Here it is shown. So your mini white is expressed in the thorax because your reporter gene must have integrated near an enhancer. The gene is involved in thorax development. Your gene here in this construct, your gene has integrated in a region where enhancer is involved in wing development. You look, the wings are showing you slightly reddish color due to mini white. Now, let's say we were interested in eye-specific genes. What people do, they have, let's say you made these 5,000 transgenic lines. Now, you were interested in eye-specific enhancers. What you will do in, normally people do, they simply isolate the imaginal discs. Imaginal discs are basically, these are pouches of cells in third instar larvae stage. Third instar larval stage. So larva in fly looks like sundi jo hoti hai. So it's like this. And here, these pouches of cells, which are fully determined to make, let's say, this is a stage where cells already are determined. So this region makes eye and the upper one makes antenna. So this is eye disc and this is antennal disc. These are antennas. And these are adult eyes. Adult eyes are made from this region. Similarly, fly will also have wing disc, the ones pouches of cells which make wing. There are leg disc, which make legs. There is halter disc. Halter makes, it's not clear here, these are little bulbs. Let me see. These are small appendages in, yeah, here it is. You can see just behind the wings, this bulb-like structure. They are important for maneuvering the flight. So after isolating imaginal disc, if you have used leg Z in your this enhancer trap construct, you stain these discs with X-gal, the substrate for leg Z, and I try to now draw the stain disc. See, this is the wing disc. This is the halter disc. You have the leg disc. Okay. You stained with X-gal. Let's see, we see blue staining, leg Z staining pattern here in the eye disc. Just in this region and nowhere in other discs. So you will say my transgene construct of enhancer trap has actually landed in a region where enhancer is present, and this enhancer is normally acting on eye-specific genes. It is an eye-specific enhancer. You isolate imaginal discs from another larva, eye disc, wing disc, halter, leg disc, etc. Let's say you see staining in eye disc of leg Z, as well as in the wing disc, but not in halter legs. You will say this is not truly eye specific because it's present in the wing as well. So you will not chase this one. This is your target. Similarly, if you are interested in wing specific, so you are looking for staining patterns in which only wing should show staining and none of the others should show staining. So that's how people have cloned, enhancers as well as promoters of those screens are called promoter traps, okay, in which you don't have. So in case of promoter trap, you don't have even minimal promoter. Here you had minimal promoter. In case of promoter traps, you will not have even minimal promoter, just your gene of interest. So people clone these enhancers, and then once you know I have this eye specific enhancer, you can simply do inverse PCR. And by inverse PCR, you can clone the flanking regions. For example, your gene of interest, this construct is here. Through inverse PCR, you will know the flanking regions of the genome, and you, in a matter of day or two, you will know, okay, this is the precise position where my reporter is landing. And then you may simply sequence. Now the whole fly genome is sequenced. You just look on both left and right side of your construct and see if there are some genes or there are some enhancer elements, and you clone that enhancer. Let's say this is your enhancer. You clone this enhancer. Now, still your job is not done. You now take this enhancer, clone it in transgene with, let's say, luciferase or GFP, and make transgenic embryos, again through pole cells. Or you can put this construct in fruit fly cells. This will be quick. And see if luciferase or GFP expression is induced or stimulated due to presence of this enhancer. If it is, tenfold enhanced, you will say, yeah, this is enhancer region. I have right enhancer region clone. And then you will, in wild types, you will see the genes on which this enhancer is acting. So this way you will know eye specific genes. You will be able to discover all the eye specific genes. So we have gone through different strategies. For example, we have seen simple EMS, MMS screens. In Arabidopsis, we said tDNA mediated mutagenesis as well. In fruit flies, P element mediated mutagenesis. In fruit flies, we also just saw enhancer trap screens for, let's say, wing specific genes or leg specific genes, and then you clone them. tDNA, P element, and these ones are easy because you can very easily map the genes and recover the mutations. Very easy. tDNA, why it's easy? You know the flanking regions, the border sequence. By simple PCR, just like the inverse PCR here, you can, within few days, know your genes. In case of classical mutagenesis where you use EMS and MMS, you have to map the chromosomes. The first thing, once you get mutant population, you cross them with wild type. And the first step is to determine the phenotype is dominant or recessive. Second step is then you map the chromosome. Where in the genome, let's say these are 1, 2, 3, 4, on which chromosome your mutation is lying. So you use different chromosomal markers. Let's say second chromosome specific markers, cross them with your mutant and look at recombinants in the next generation to see if I am lying on the second chromosome or third, where my mutant phenotype is co-segregating, because mutant marker is also the known mutant marker should co-segregate with your phenotype if it recombines. In fruit flies, there are chromosomal balancers. So your today's assignment is chromosomal balancers in fruit flies. What are chromosomal balancers in fruit flies and what is their use? That is your assignment. So using these chromosomal balancers, you can very easily, if your mutations are lethal, you can very easily determine whether your mutant phenotype is segregating due to mutation on second chromosome or third or fourth, etc. And based on the phenotype, also you can group them, as I said, due to their co-segregation and recombination with known chromosome markers. Once you determine your mutation is recessive, let's say your phenotypes are recessive, another thing you should do is to determine if mutations are recessive. What you do next is you analyze them through complementation. We will talk in detail in next lecture. Why we do complementation? Let's say we have discovered 50 different mutants, independent mutants. Now, out of these 50 mutants, is it possible we have 5 mutants which are containing mutations in the same gene? A gene A, let's say this is gene A, and this is in mutant 1, mutant 2, gene A, mutant 3, 4, 5. In mutant 1, gene A is mutated here. In mutant 2, it's mutated here. In 3, it's mutated here. In 4, it's here. And in 5, it's a different. So these are possible. And these are basically when same gene is mutated. It is different form of the same gene. And what do we call this? When same gene exists in different forms?
Alleles.
These are alleles. So these are alleles of A. Now, out of 50, how do you know I carry 5 mutants within the same gene? So the whole purpose of genetic screen is to be very smart and minimize your work and exclude, instead of saying I have 50 independent genes, you may have 10 independent genes and all the others are out of 50 are alleles of different genes. Okay. And this is easily resolvable. This is doable through complementation, but the condition is through complementation, we can find alleles of different genes if the phenotypes are recessive. So all the recessive phenotypes, what we do, we cross them with one another and then look for complementation. So we'll talk about this in next lecture. How long is this lecture? I think by 10:45. Oh, no. We have 8 more minutes because I started. So what is complementation? Let me quickly give you an idea so that when you go back and read, so look through these slides. When you come back, you will, in next lecture, understand complementation test better. But just as a quick help, I explain you. So let's say we have I phenotype. Eyes are white. It's recessive. This is our mutant 1. Mutant 10 is also eyes are white in fly, and it's also recessive. So what we do, we cross mutant 1 crossed with mutant 2, and then we look for eyes. There are 2 possibilities. One possibility is the F1, it shows white eyes. This is first possibility. The other possibility is the F1 progeny shows red eyes. So what is happening? Can you guess?
सर F1 जहाँ पे white eyes थी वहाँ पर complementation हो रही है.
जहाँ पे white eyes हैं यहाँ पे complementation हो रही है.
जी सर.
और यहाँ पे?
सर यहाँ पर different alleles है.
यहाँ पर different alleles हैं. Alleles of what? Same gene?
Alleles of the same gene, yes.
Anyone else? कौन किसने आंसर दिया था ये?
Sir, Amna.
Amna, can you switch on your, so now since lecture is coming to an end, please switch on your cameras. Everyone, please switch on your cameras. So here, when it's white, complementation is not taking place. When it's red, complementation is taking place. They both are recessive. If they are alleles like this of the same gene and they remain white, it means complementation is not taking place. And if eyes turn out to be red, it means these mutant 1 and mutant 2, they are mutants in different genes. And now complementation is taking place. So you should go back and read about this. I stop it here. So I gave you assignment about chromosomal balances in fruit flies. And second is this complementation. And Najma, have they done the recitation time? Finalize हो गया?
जी सर वो Friday 2:30 to 3:30 हुआ था.
Yeah. Are you doing some genetic problems with them?
Yes sir, so they were saying कि अगर हम assignment के questions को ही solve कर लेंगे तो उससे हमारी help हो जाएगी. So I answered their questions in the recitation. But they have not yet confirmed office hours other than the recitation time, which I want them to let me know.
So you should you should all, be more active, because this course will slip out of your hand if you don't do problem solving in recitations and the assignments. Don't think that the assignments we are giving you, it will help you. Najma has to help you with Najma, आपने प्रॉब्लम सेट्स उधर एक्सप्लेन करनी है जेनेटिक मैप्स की और कॉम्प्लीमेंटेशन की, एपिस्टेसिस की, क्योंकि ये अब ये जब हम फॉरवर्ड जेनेटिक स्क्रीन कर रहे हैं तो इसमें अब आगे कॉम्प्लीमेंटेशन के आ जाने हैं.
Yes, sir.
And then we are going to next level when we'll talk about different pathways affecting the same different genes affecting the same pathway, then epistasis will come. So the more problems they solve, the better they become. Their understanding and concepts will become better. So all of you should go back and also pay attention about this complementation, so that next lecture, when we talk about this yeast story or fruit fly story, you should be able to understand this. Okay? Any question? So if there are no more questions, thank you very much. Have a nice day there.